I'm really happy to welcome Ritu Das. This was our second day, so as Mike said, welcome to ISPASS Day 2. Ritu is a associate professor at the University of Michigan, and she'll be talking a little bit about systems for precision health, but I wanted to first introduce her. So she's been doing lots of fun projects, so in-memory architectures, custom compute for things like precision health and AI, which we'll see a little bit more of today, but also other things, fine-grained heterogeneous core architectures, low power interconnects. This is going back probably kilocore processors, right? She's authored over 45 papers, filed 7 patents, served on over 30 technical program committees, and she's received 2 IEEE Topics Awards, NSF Career Award, CRAW Borg's Early Career Award, Intel Outstanding Researcher Award, Sloan Foundation Fellowship, and she's been inducted into the IEEE ACM Micro and ISCA Hall of Fame. So with that, I'm glad to welcome Ritu, and can't wait to hear more about the systems for precision health.
Thank you, Trevor. Thank you for listening to me, folks. I'm so excited to share our recent research with you and generally give you a perspective of this very exciting area. So I'll be talking about systems for precision health. And before understanding systems for it, I would like to just give a very little brief overview of what precision health really is. So precision health is a broad way of treating diseases by using the unique characteristics of each individual. Because you and me, we are all so different, right? So, so looking at that variability in genes, environment, and lifestyle is the key to precision health. And that can reap a lot of rewards in terms of treatment and diagnosis. So this initiative was started about 6 years back by President Obama. And he makes a very nice argument that blood transfusion was a very unique and important discovery. And for blood transfusion, you had to match the blood types of a person. And imagine if you don't do that, what would happen, right? And if that kind of precision was needed for such a critical treatment, why can't we bring that precision into every kind of treatment we do for curing cancer, for treating all other kinds of chronic diseases? So this initiative was started by about 6 years back and backed by NIH. And a lot of interesting research has been going on in the space outside the systems and also in systems. So the basic premise of precision health is that one size doesn't fit all. You can't take one prescription, one drug, and then give it to en masses. If we could customize it for individuals and their lifestyle, their variabilities in their sort of genetic makeup and epigenetics, that is what precision medicine is about. So now that is the vision. Let's see how a typical precision health platform looks like. And this is a very generic platform, but so many of my projects, I've seen the same pattern repeat often and often across different diseases, across different risks, across different diagnoses. So I'll make this as broad as possible. So precision health platforms start with data sources, some kinds of unique data relevant to the patient, starting with electronic health records. This gives you the patient history, then it's the patient's genome and their genetics, their genes, how the genes are being transcribed. So that is the epigenome, and how they are actually converting into proteins and RNA and how that makes up your body. Then also an interesting part of this is the microbiome. So our human body consists of trillions of microorganisms. In fact, these microorganisms outnumber human cells by 10:1, and they play a vital role in human health. So, all the bacteria inside you, that plays a big role. And so that's another input in our data sources. And then upcoming is metabolome. Metabolome is basically the biochemistry inside you, the small molecules, chemical molecules inside your body in biological samples, like lipids, sugar, amino acids, and fatty acids, etc. Lots of them, like hundreds and hundreds of them. So we take all these data sources, we have to have a way of digitizing them, lots of data. And just to put into perspective how much data we have here is a Facebook account would have about 0.5 gigabyte of data per user. But if you do liquid biopsy, that's, Grail has reported terabytes or several hundred, one genome data, after you do the replication, etc., could be 300 gigabytes. And for liquid biopsy, it would make terabytes of data. So that's like 1,000x higher magnitude of data processing we're talking about here. So you take all this data, and one important step here is sequencing. And why do we need sequencing? Because some of our signals are raw biological samples, right? Raw biological signals. These are chemical molecules, base pairs. So how do we, proteins, how do we kind of translate that to a digitized format, which our models can interpret? We have to go through a sequencing step where we convert these into a digitized format. And so that's a big part of this puzzle. And then using the sequence data, and we do a lot of analytics over it and analysis over it, have standard pipelines, standard tool sets, and evolving ones too. And together I call this as decision support tools, which would interpret all this data. And finally, after interpreting the data, the outcomes are clinical decisions. So the clinical decisions would be either diagnosis and prevention, or treatment and management, where treatment could be, specific drug type, or lifestyle changes, nutrition changes, what kind of activity you bake into your life. And so that's basically a broad stroke precision health platform. Always this downstream. Now, the number of data sources might vary, the analytics pipelines used might vary, but pretty much this is the template. So now let's take a look at what are the system considerations for precision health. Efficiency, we are computer architects, right? We care a lot about efficiency. It has to be energy efficient. It has to be really fast, ultra rapid. That's where we can show our powers, right? By acceleration. And storage cost is also very important. So managing, in fact, storage plays a big role in even efficiency. So having better storage devices, being able to compress data, reducing that cost is pretty critical. Security and privacy is another very important aspect of precision health systems, because you are exchanging, so by definition, it is giving unique data of a person. So it's kind of easy to reverse engineer things. And people care about privacy of all the data which they're giving. So, having some kind of secure system like an enclave or homomorphic encryption to process that data without giving away privacy is also a critical design consideration. And then form factor, right? So we want to make it real time. A DNA pioneer, Churchill, he said that, won't it be cool that you're just walking around, you fall sick, you sneeze into a device, and the device would tell you what viruses you have and what bugs you have, and give you a prescription plan. So that's the vision. And actually, we're getting quite close to it because we have handheld sequencers today. So form factor is very important. And now, as the sequencing devices form factors are shrinking and becoming smaller and smaller, it would be nice to have edge compute devices which have very small factor, which can integrate with these sequencing devices. So sort of these three pillars guide the system design of precision health platform. What we will focus today is in our decision support school on sequencing and its exploding applications. And also I will show you many end-to-end applications using sequencing. So sequencing is the key ingredient of precision health. Now, how has sequencing, what's the history of sequencing, where we were and where we are now? The very first sequencing, it was somewhere in 2001, very primitive sequencing took like a lot of time, many years, decades, in fact, to get to that first genome. And then slowly we see like a ramp up, there's a knee of the curve, and then it started ramping up quite a bit. We got the 1,000 genomes around 2012 or so. And then that was the that was the second generation of sequencing after the first, that was before the 1,000 Genomes project was second generation. Illumina came up with its own machine. And then now we have third generation of sequencing right around 2012 or so. These are really portable, high throughput devices. They have reduced from the size of a refrigerator to a washing machine to something which you can hold in your palm, like a USB stick. So that's where we are. And it's actually ramping up quite a bit. And in fact, later, so before we had this notion that every person has only one DNA representing them, a DNA string, but now that notion is exploding in the sense that single cell sequencing techniques have been producing, because every cell inside it has its nuclei and DNA, and every cell is unique. The liver cell is different from the heart cell, and skin cells and fat cells are different. So now we see an explosion there where we can do single cell sequencing and even get more precision into the diagnosis. So that's what is happening to the sequencing technology. It has been ramping up quite a bit in terms of throughput and the number of genomes we are able to sequence. In fact, countries like UK are thinking by 2025, they're going to sequence every person, each one of their citizens, okay? So that's exponential growth in sequencing. Let's see what is happening to the cost. The very first genome took about $100 million. Like, that's crazy. But what is really cool is look at this graph and where it has shooted. In fact, today we can sequence in our lab with about $100 to $200. So it's really taken off. It's really starting to become cheap. And in fact, as the volume increases, the demand increases, then it's going to drive it down even cheaper, because the raw materials for these sequencing devices are not that expensive. So we need a high market volume to drive that cost even further down to tens of dollars. But as you know, no computer architecture keynote can go without a Moore's law. So what we see here is, as the sequencing industry is really, rocking it and really going down to that low cost, our servers, our edge devices are not able to keep up with that compute throughput. And today we are not sequencing that many genomes. I mean, still a lot, but imagine if every year, like when you do a blood test, you have to do your genome sequencing just to know, like to do a liquid biopsy or something. And that's like exploding amount of computation there, right? And not just that, you're hospitalized and you want to keep sequencing the pathogens and bugs inside your body. So with all that sequencing going on and all that precision health platforms blooming and growing, we need computer systems to keep up with that growth. I would give you a little preview of exploding sequencing applications. And we started looking into this about a few years back, maybe 2 or 3. And we had no idea how much this field is exploding. So for instance, the recent and very close to our hearts, and we have all been suffering, is the COVID-19 pandemic. So when some virus is going around and causing a lot of loss, and it brought the world to a halt, you want to know what virus it is, what strains it has, how the epidemiology of the virus is developing, and how the contacts, it can help you do a more precise contact tracing. Only if we could make sequencing cheap, we could have even avoided the delay and the lag in testing of the pandemic. And I have a project which I'll show you how that could have been prevented. Consumer genotyping, many of us might be familiar with this 23andMe. We want to know our ancestry, our roots, what kind of disease we might be at risk to. So this got quite popular. Then for cancer treatment, precision health and using molecular information. So when I say molecular, it is DNA or RNA. Understanding that molecular precise information is very important. So in Michigan, we have so many drug trials going on for cancer, and you want to track it. You want to track the progress without necessarily doing an incision into a brain every time to know if the tumor is shrinking or not. And to track that progress, basically you can take a sample out of your spine or your blood, and then you can track how much of cancer cells are floating around. And that is very, very helpful for tumors which are in invasive places, which cannot be, where biopsy can be expensive and not feasible. For microbiome, this is kind of microbiome, the more and more I get into this field, I get more and more excited about microbiome because it really defines so much of you. And it's one of the areas in this field which has been not explored that much, and it's just a lot of attention and research is going on to it. And NIH has announced a microbiome initiative where they want to study this, track this, and see how it affects your gut health and your overall well-being. Microbiome is, and you have sequencing which can actually tell you the microbiome composition. I'll show you a project which does that. Then we have got, it's proliferating into agricultural sequencing. You want to know the crop health and the non-genetically modified crop. How does it do? How does it, which variety would give you a better yield? And another cool and exciting application of sequencing is tissues from your brain or from your breast or from your lungs. Then you want to know the margins. You want to know how much of it is cancer, how much of it is not cancer. And won't it be cool if we can do sequencing within 20 minutes and tell you that? Food safety, all through the supply chain, we can do sequencing and figure out where, at what point the food got impurities and got polluted with bugs, so that we don't have to throw away all that amount of food. And there's a very cool example of Chipotle, how it had to throw away tons of chicken. But if they had like a metagenomics analysis, they could have saved all that money and all that hard work which has gone into producing that. And liquid biopsy, of course, that's my favorite example. And that would be amazing. So many people are being affected by cancer. If we could just do a blood test or some simple non-intrusive test every year and do an early detection, a lot of lives could be saved. And liquid biopsy is like exabytes of data. So that's a nice computational problem too. And having portable pathogen detections to detect what kind of diseases and bugs we have in our body. So lots and lots of cool applications. The sequencing technology itself has evolved quite a bit. Illumina started producing sequencing by synthesis, where they create, basically it's like a luminescence, and they create these bridge amplification of molecules, and then by studying the light intensity and the color of light, they're able to do figure out the base pairs and base call for a DNA. So 1000x increasing in machine throughput. But now what's even more fascinating is nanopore sequencing, which is real time and portable. It has increased the read length. Read length is just part of the DNA which you're able to sequence at one go. Quite a bit, and that's very exciting for biologists because it can tell you a lot more about your genetic makeup. But the per base inaccuracy, the accuracy has dropped, so it is quite inaccurate. And but that again is exciting for us because we can informatically solve that challenge of inaccuracy. And it's real time, it's streaming. So let's look a little. So what I think of Illumina is a second generation, and this is third generation. So let's take a little bit of look at what nanopore sequencing really does, and why it is real time, and why it is so small. So what nanopore has done is basically created these biological pores. So if you open up a nanopore device, It has got hundreds and hundreds of flow cells. Inside the flow cells, they're just basically biological membranes or pores, which suck in your DNA. And this sort of graphic shows you that how DNA gets split and then sucked in through the motor protein into the nanopore. That's where it gets its name. And and what we do is, now here's where our computer engineering background helps us. So in the spore, when the strand is going through, it creates different voltages. And that can be read out using ADC to create something called squiggles. And then a base caller algorithm can translate those squiggles into base pairs of DNA. It's massively parallel. There are thousands of parallel pores embedded into a flow cell, and you can get billions of base pairs out of a nanopore. So, I had a very quick video to show you about nanopore. I thought this was fun and it would give you some background into this. So let's take a look.
Holes that in nature form gateways across membranes. In our technology, protein nanopores are embedded into a synthetic membrane, bathed in an electrophysiological solution, and an ionic current is passed through the nanopores. As molecules such as DNA or RNA move through the nanopores, they cause disruption in the current. This signal can be analyzed in real time to determine the sequence of bases in the strands of DNA or RNA passing through the pore. During sequencing, the nanopore analyzes the entire fragment of
So it's real time streaming output. So for Illumina, it's very expensive. We have to batch, we have to wait a week to get all these genome sequence. But this is like carrying a USB stick. You can take it into the field. Here are the pictures where they are sequencing Zika virus in the field, and this is in a space station. So it's portable, you can carry it. It has got streaming output, really low cost. Each device is only $1,000, and that's going to go down. In contrast, an Illumina machine would be several tens of thousands or hundreds of thousands of dollars. It can do both RNA and DNA sequencing. So it can sequence human genome, it can sequence proteins, sequence the RNAs which are derived from the DNA, and it can sequence the bugs, bacteria and virus. So this is a fascinating device which has opened up a lot of real-time applications. And we'll talk about some today. So, we went ahead and created a nanopore sequencing lab at Michigan, and that's Jack there sitting, actually sequencing stuff with our wet lab. It is a biosafety level 2 certification. We can do both tissue and RNA, and I'll show you some projects where we actually do the whole end-to-end thing from tissue to the diagnosis. So this was a lot of fun for us, and it really helped us do a very deep, tight coupled co-design research between this edge device. You can think of this as a sensor almost. It's an edge sensor, and the computing algorithms and software and the hardware architecture which goes on top of it. So that gave us a good overview of sequencing technologies. Now let's take a look at some end-to-end application case studies, just to see what we as computer architects can do to help this field. So the first end-to-end application case study I would look at is whole genome sequencing. This is where we started. So whole genome sequencing, the process is we get a sample, okay? It could be from any part of your body. And this sample is prepared and sent through a sequencer. And what this process sequencing does is, you can think of human genome as a very, very large string with 6 billion characters. The sequencer snips out this very long string and chops it into many tiny, tiny strings. In fact, it replicates many parts of it. So now you have to basically solve a giant jigsaw puzzle using these tiny reads and put it together to recreate the original DNA, the long string. And that is done using several steps. We do read alignment, where we take these small reads. Read is a fragment or a shorter string of DNA. And we try to map it to the most matching part in a reference genome. So this could be a reference genome of, which is kept standard across most humans, and a specific human is aligned to this specific reference genome. So that's a read alignment step, and BWA-MEM is used for that. It's an industry standard, mostly. And then we have got the part where we, once we have aligned it, there is some bookkeeping to do, post-processing. We do sorting and marking duplicates. This is just simply sorting the reads based on the reference genome position. And then there's a very critical step of finding out the variants. This is really the heart. What we want to do with whole genome sequencing is find those mutations, find where we differ or where we have variants from the reference genome. And so that's usually using some statistical models like pairHMM and so on. The tool used for this is GATKey haplotype caller. And after we have called the variants, then we send it to the physician and diagnosis is done. So if we run this on a pretty large CPU, server CPU, takes about 14 hours, or a CPU with 32 CPUs, so that's about 400 CPU hours. And you can see the breakdown of time here. A lot of time is taken in seeding and seed extension, which are part of the read alignment. And then we have got sorting and marked duplicates. pairHMM is another key component of it. And then there are some other parts which, other intermediate steps, which I have not shown here. So what we started out with is attacking these bottlenecks one by one to get an end-to-end speed up. First, our work was read alignment, GenAx. The whole idea there was to, and read alignment is usually done using a dynamic programming approach, where you basically fill out a DP function for a cells by taking a read and figuring out which parts of the reference match to it most. And the path through the dynamic programming box, which gives you the most optimal path, is the alignment, which we refer to. So this, our approach but very quickly we found that that was too, it lacked some features, and for automata to work, you needed to program every string in the reference into the automata. So that's 6 billion strings, and that's too much. The reconfiguration time was too bad. So we ended up creating our own very customized automata. We call it string independent local Levenstein automata. The string independence comes by just doing a NOR function between the base pairs and having a retro comparison. So, and this basically the hardware architecture for this, the accelerate architecture, resulted in a systolic array which could implement this automata. And this, we fabricated a chip for this, and we demonstrated its efficiency, and it's about 63x better than a CPU solution running dynamic programming. We further went ahead and also improved this further to make it more area efficient, because we wanted to have an end-to-end system. And also we realized that we are better off running these accelerators on an FPGA because the ASIC requires much higher market volume, and that may not be the case today. It may be the case tomorrow. So for an FPGA solution, we are quite area constrained. And so we came up with a speculation and check mechanism. So the key concept here is, like, when you are building this DP matrix, there's the full band will give you the optimal path, but most parts of the full band are not utilized well. They have low utilization. The cells in that doesn't really matter. You could just set that to be 0 and your answer will not change. So we thought if we could do a narrow band, that would be awesome, because usually the number of cycles and P's is proportional to how many cells we are computing. So in a narrow band, all this white space is not computed. However, the caveat there is that what if the optimal path is outside the band? You might miss it. So to solve this problem, to get optimality and area efficiency, we developed a speculation and check approach, where you speculatively execute with a narrow band, and we computed mathematically threshold scores which can guarantee that a path found by the narrow band is optimal or not. by checking against these threshold scores. And we had some cool optimizations about, how we can, these threshold scores, we can also do using an edit machine, which is much more simpler, very tiny, like 8 gates. So that was the SeedEx check algorithm and accelerator, which again helped us gain, with 100% equivalent results, it helped us gain a 2x area efficiency and a 6x higher throughput. Another key bottleneck with genome alignment is seeding part, where you have to take the initial reads and figure out which spots in the very long string does it match to. After you figure out those spots, then you go and align it using the Bandit-Smith-Waterman algorithm I just showed previously. And this is very memory bandwidth intensive, as we are showing here. Gets like kilobytes of data per read. And the reason why it is very memory intensive, you can think of it as a very compressed index, like a hash table, but very, very compressed. And so to go and fetch that, you and it's pretty big too. It's about many gigabytes, so it doesn't fit in your cache. And it has very poor locality because it is so compressed. So you spend lots of bytes fetching this data from memory. Our solution was to create a bandwidth efficient data structure. And this is a very good example of hardware software co-design, because this bandwidth bottleneck is something which we architects can see through and think of how to get over it, right? By improving the locality. While the bioinformatics people only wanted to make it smaller and smaller, and they didn't realize that's going to make it very bandwidth inefficient. So our solution called enumerated radix tree, basically, it has several concepts in it. But the key there is kind of decompressing it, trading off memory capacity for memory bandwidth. So it is larger, but it can do multiple of these index lookups in one go. And using something called suffix tree, a radix tree. And it has got pretty cool optimizations to reduce the maximal match phases. It does a zigzag MEM search to reduce that footprint. So that together, enumerated radix tree and SeedEx, the latest design, basically made read alignment quite fast for us. And on an FPGA, which we have deployed on AWS cloud, it can run. It's about 2.3x. Just the software itself is about 2x faster than BWA-MEM. And so we started out with, but our software was also fast, and so it got incorporated into make even software contributions by understanding the hardware bottlenecks better. And then we also accelerated the sorting and mark duplicate space. Nothing fancy here. It was just an in-memory sorting approach, which gave us tremendous amount of speed up. But we wanted to do the end-to-end pipeline, so we solved this stage too. The last stage for the variant calling, we took a pruning approach where we figured out that, in variant calling also it's a dynamic programming algorithm, but a lot of the cells which we compute there don't really matter to the end result. So what we could do is we could compute this in a lower precision and then figure out which cells matter and do it in a higher precision, and then compare the two results with some bounds and checks so that we can do the pruning and still guarantee bit equivalence and optimal results. We also built a chip for this. And so for the whole genome sequencing, we have built chips and FPGA prototypes which are actually working. And throughout the theme of our whole genome sequencing research, we wanted to make sure it was bit equivalent and accurate. The reason for that is, human genomes really differ by very small. Here is Hillary Clinton and Trump. Their genomes probably differ by 0.1%. Human and chimpanzee differ by only 4%. So if you don't want to call a human a chimpanzee, then you better be accurate with your whole genome sequencing. So it's very important to focus on accuracy while you're doing all these optimizations. And so we did approximation, we did pruning, but we made sure that the path we are still finding was the most optimal path. So I'll show you a quick demo by my student Arun for the CYP2C gene. So
in this demo, we show how ERT and SeedEx can be used to align reads about twice as fast as software BWA-MEM2 and call variants in the CYP2C19 gene, widely responsible for drug metabolism. After downloading the alignment outputs, we now move to the location of the CYP2C19 gene, the chromosome 10. Each red block corresponds to an aligned read. We can see that more than 50% of the reads have a G to A variant in this gene.
Okay, so that was a demo on an FPGA. And so why this gene is important? So this basically involved in metabolism of common prescribed drugs. So if you have a normal CYP2C gene, your DNA will transcribe and create a long enough after splicing, after cutting away the non-coding genes, a long enough RNA which will help metabolize drugs like Plavix. But if you have an aberrant gene, then a tiny flip here could cut away a long piece of even the coding sections, which can actually create the proteins and create an aberrant RNA. And that would lead to poor metabolization of the drug. So understanding this gene is important. And there's several such examples. We could show you this in a picture. So we showed you this aberrant. But that is why finding out these variants are so critical to understand all the way from critical diseases like cancer to understanding metabolization of drugs like Plavix, which is a common drug, blood thinner, prescribed to many diabetics and heart patients. So in our acceleration case study, we started at about 14 hours on 32 CPUs. And for an FPGA system on cloud, we have all these components on the cloud. We could reduce it to about 2.5 hours. An FPGA, you can use it today, right? You don't need to build a very fancy $1 million chip for that. But if you could go to ASIC, then we could reduce it to 2 hours. What you would notice here is that there's a big other portion, which is a non-accelerated portion. So that part can be accelerated further. So that's where Amdahl's law kind of, bounded our speed up to 7.3x. Other portion has some components which we have still not attacked. So that's also interesting research left to be done. But those could be accelerated to get even higher speedups. But we did go end to end and we did look at different bottlenecks. Now I will do one more case study, and this will be pretty quick, but I'm so excited about this. This is ultra rapid cancer diagnosis, and let's see what this is. So the whole idea here is to do intra-operative molecular diagnostics. So when you are on the operation table, we take a tissue and tell you the tissue has cancer or not. So what's the use of it? So today what they do is they cut out a tissue from, let's say we are doing a brain surgery, and they take a slice of it, a very thin slice of it, and look it up in a microscope, which is called histology, like a very fancy microscope. So it's quite primitive. There's a manual, there's a person sitting there looking under a microscope and telling you whether this tissue has cancer or not. It would be fantastic if we could augment this to genetic information and actually tell you whether this tissue has molecular variants, which are biomarkers, which are cancer causing. So basically the whole key here is to make it really rapid molecular sequencing that we can replace this age-old methods of microscopy. So can we sequence a tumor's DNA within intraoperative time frame? That was a challenge we undertook about a year or so back. So how does sequencing-based diagnostic work? So this is the pipeline which we created. You take the tissue, you do DNA extraction, then you do gene target amplification. So you look at the specific gene, which has the biomolecular, and then you do some library preparation so that this DNA is ready to be sequenced by our sequencer, a portable sequencer, and then we do the informatics. So the target amplification is done by a PCR. Now with COVID, we all know what PCR reaction is, but it exponentially amplifies a specific region of the target. And this, just an unoptimized design. So Illumina cannot do this, but with Nanopore, at least we can do it in 2 hours. This is still quite slow. So basically, we developed a threshold sequencing mechanism which would co-optimize amplification time with sequencing time. The whole idea is to reach to an optimal amplification time where we can go and sequence. There's just enough amount of amplification of that gene chemically using a chain reaction that we can go and sequence and informatically get that information. So since we are able to after correct with sequencing, we don't need to go all the way and spend several 1 or 2 hours amplifying the product. And this is a very cool marriage of bioinformatics along with wet lab. And by doing that, we managed to get it to 52 minutes using a PCR reaction. And that was still very thrilling. And it was the world's first demonstration of a sub 1 hour sequencing. But 52 minutes is not enough for an intra-operative sequencing where the patient is open on the table. So next we looked LAMP, which is isothermal. It's a loop-mediated isothermal amplification. PCR, the reason why it takes a lot of time is because we need to keep changing the temperature high and low. And that's a mechanical action. It has to take some it's physics. You can't reduce it beyond a point. But LAMP, because it is isothermal, it operates at the same time. It is, you can very quickly amplify things. But the problem with LAMP is that it creates a lot of spurious products, a lot of bad amplified product, which we need to correct. And so we created a tool, LAMPRAY, which is open sourced, and it can find all these spurious products and glean out the needle in the haystack, which is our target. And what this tool helps us do is it reduces the sequencing time by half by recovering more than half of the product. And that also reduces the amplification time. So LAMP by itself would be very hard to use here. But with our tool, we can make that happen. And we could get it to under 30 minutes or about 25 minutes. So we started with the baseline protocol, which was above 2 hours. Well, Illumina is like days. So this is 2 hours. And then we got to LAMP threshold sequencing, which sets the world record for fastest time to result for cancer diagnosis. And this is all being done in our lab. Jack is doing this end to end. He has a timer. He takes the tissue from the tumor and gets to the mutation, hotspot mutation for brain cancer. So that was very exciting. I'm going to do a very quick overview of acceleration study for real-time pathogen because we're running out of time. But here the whole goal is, I don't need to motivate this. Pandemic is out there. We need to detect viruses and we need to detect superbugs. These are antibiotic resistance bacteria, and they are touted. They are saying that superbugs will kill more than cancer by 2050. So it's a huge problem which is coming up. So now we are seeing pandemic. The superbug pandemic is coming up, I assure you, unless we do something about it. So, to test COVID, it took us months because creating a PCR design is very complex, right? So how can we prepare for the next pandemic is by creating a universal pathogen detector, which can detect anything. So immediately a pandemic happens, you detect this pathogen, distribute the electronic signatures of that pathogen through an email. You don't even need to send all these solutions all over the world. You just email out the electronic signatures and then it's digitally distributed. And then you can sequence it to know if there's a pathogen or not. So that's like ultra fast mobility of a pandemic diagnosis and detect. And if we had done this, if we could have done it for this pandemic, it wouldn't have spread so much. How we can go by this is, we can use something like a real-time sequencer like Nanopore, where we can send it through a base caller, and then there's an accelerated filter because a lot of these, we're getting this DNA from a nose swab or your blood, which has a lot of host DNA, and we want to throw that out. We want to discard that. We just want to keep the bug DNA. So we need an accelerated filter, then we can do the analysis and interpret it. So this accelerated filter is what we looked at, and the sequence analysis. So the cool part of Nanopore is that it is streaming. So when you're designing this filter, you can actually tell Nanopore to discard the rest of the read. So it's a real-time feedback to the device, which is read until. And also when you have reached enough confidence in your result, you can just stop. So while in Illumina, you need to do the whole many-hour run, right? You cannot just stop arbitrarily. So this gives you quite fast detection. We took it a step further and skipped the entire base caller, so we can directly work on the squiggles and need not call the bases. And that Pathfinder ASIC can be integrated into MinION, and it can do a SARS-CoV genome under 2 hours. And it helps reduce sequencing time and cost by about 40%. And it's very, very much efficient over a GPU and CPU. And this is also for the microbiome. So that was for detecting viruses, but let's say you want to understand the microbiome composition. So this is a run we did in our lab with DNA extraction. And this is the ideal output, which we know from the composition. Like these are the different bacteria we have. This is the pie chart we were expecting. And this is the pie chart we produced in our lab for about $90 and in 1 hour. And you can see that they are very close to each other. It's very hard to find a difference between these two pie charts. So we can do a true abundance estimation, which is unbiased without amplification using the same sequencing pipeline, which I showed you. So, so how can you kickstart precision health research? Well, please feel free to download the genomics bench, which we released in ISPASS this year. And it has 12 computationally intensive kernels, covers major steps, and gives you a lot of wealth of information about these sequence analysis pipelines, includes the data set. I hope you check it out. And with that, I would conclude the talk. Our team consists of, it's a multidisciplinary team with experts. Satish and David are my close collaborators in the CS, and Jenna, she's a machine learning expert. And we have got Bob Dixon and Carl Koshman, which have been helping us a lot with microbiome and pathology and brain cancer. Work has been done by an awesome group of fantastic students. I am flabbergasted by their enthusiasm. Arun, Daichi, Jack, Zhao, Timothy, Hari, and Yufeng, they're just simply awesome. They don't stop at taking up any challenge. They want to, they want to understand biology, computer science, hardware, architecture, everything. I love them. Thank you, students, for awesome work. That's it. Thank you. I'm happy to take any questions.
Great, Raju. Thanks so much. I'm going to clap for you. There's some other people I see clapping too. It's difficult in the, pandemic times. We got some claps, so that's really good.
Thank you.
And we've got some virtual claps as well, so that's great. So now we have a few minutes where we can open up to questions. And what I would encourage people to do is, if they have any questions, feel free to, send them off in the chat, and I will facilitate those questions and we can ask Ritu and see what her feedback is. But until we get some more questions, what I will do is I have a couple questions that I've been, thinking about since I've been looking at. I used to do a little bit with bioinformatics back in the day. So there's a few interesting questions I have here for you. And so just feel free, drop some questions and we'll move on from there. Okay, cool. So let's start. So, we in the very beginning of your talk, you started talking about like exabytes of data and wow, this is a cool application. But does the healthcare industry have the ability to support terabytes per person for this liquid biopsy that you're talking about, right? Are we ready for this yet? Or is this a special case? Because it seems to me like, we're just getting digital health records. So I'm wondering, is this like, maybe we can sort of do this at the same time while we're rolling it out? Like, what do you think is happening?
Yeah, no, I think the answer is we're getting ready. So we're not ready, and we're not absolutely primitive. There are startups and like this Grail, I guess, that's a liquid biopsy company. So, storage is a big problem, like terabytes of data, processing it also is a big problem. So I feel like, we're getting there. We're still not yet there. And by accelerating compute and figuring out clever storage solutions, we'll definitely get there. There's a lot of promise in terms of results, how the motivation is there. The doctors and biologists have shown that this is useful. So it's up to us to now make this happen, I guess.
So I guess that's sort of the follow-on question, which is, how useful is this, right? Obviously, you've done a good job convincing us that, we can, having these precision health methods, can help us more quickly diagnose cancer and things like that, right? But with this information, right, have the doctors proven, for example, that having this, for example, say you're doing brain surgery, right? And we're looking at the cancer area, right? So I'm guessing the main reason why you want to do that is to make sure that you got the sufficient amount of cancer removed from the brain, right? When you're taking out, and you want to look around, you say, okay, do we have everything? Is everything looking okay? And then you do a sequence, a double check, make sure everything's good. You're like, okay, we're good to go, right? So that's a clear one, right? But are there other
things which have been proven?
So right, and that was clear. I can see it, right? But these other things aren't as clear. So I just didn't know what you thought.
Yeah, no. I think that's a great question, Trevor. So you're asking whether, like, sort of the doctors and biologists proven the use of this, and you are kind of convinced for the brain surgery thing. For liquid biopsy, I can give you an example. So we have a collaborator, Carl, whose picture I had showed, and the mutations, the hotspot mutations which we looked at, I keep forgetting this is H3F3A-K27M. So we have been trying to do this sequence in our lab and find these mutations. And what he has shown in his clinical trial for glaucoma-like patients, glioma patients with very rare cancer, especially kids, he has shown over 6-month trials that tracking this in the spinal fluids with the drug trial, they actually see the drop. So the drug trial is effective and the liquid biopsy is tracking it.
So they can say, okay, this is how fast it's helping, this is the rate. And I guess in the past, we haven't been able to see those rates of reduction, right? It's sort of like, okay, it's going to take a day to do the processing or a few days. So you really only see that.
Not just a day, Trevor, like we have to do biopsy. You have to put a poke inside, make a hole in your skull, take out the tissue, and then look it under a microscope. So liquid biopsy, you just puncture and get a little bit of fluid and do it. And these are tiny, small kids, like,
Right. You want to be able to do it quickly. So we have some questions here from the audience.
Sure.
So let's see here. Identifying the deltas helps to accelerate individual steps. So is there an opportunity to use deltas across the pipelines instead of just passing the full sequence downstream? Maybe you can pass those deltas down to the task. Have you thought or seen sort of this processing of deltas throughout the whole pipeline?
Yeah, so the deltas, you're talking about variants, I guess. So
well, it's just, I'm guessing the question is dealing with, so for example, you've got, just small changes, right? You only have small changes in the sequence. You only have small changes in the different components, right? So, okay, the proteome is slightly different. We don't need to pass the whole thing, but the downstream processing needs to be able to take advantage of the fact that we're only giving it the delta, right? So naively, I would guess that downstream processing wouldn't be compatible directly with this, right? Especially your ASICs, right? And then the question is, do you think that, I'm just extrapolating here, but do you think that if we extended these deltas, right? So for example, could we then speed things up even more, right? You're talking about sub 30 seconds, but maybe we can go down to like sub 30 minutes, but maybe we can go down to 20 minutes if we know, okay,
right.
No, we don't have to do the PCR or equivalent test, but maybe the back end, we can eliminate the vast majority of that.
Yeah, absolutely. So now also, downstream, so the human genome is several gigabytes, like 30 gigabytes, a whole sequence. But downstream to the diagnosis pipeline, we usually pass the variants, which is a VCF file, which is much smaller. So, that's, and even for like population genomics, usually these variants is what we analyze. We don't necessarily analyze the whole genome. So there is that sort of phase by phase reduction of data. And also, I think sometimes we can just get away with genotyping. I think that's what you're referring to mostly. Like we just look at specific genes and not the whole genome, not the non-coding region. So that also helps quite a bit. Yeah.
Okay, cool. So we have another question here. Great talk. Thank you so much. So any guidance to further reduce the intra-operation sequence-based diagnostics? Impossible, you know. I mean, you're already down to 30 minutes, so, you know, why not go a little bit more?
So physics, I guess. But
I'm sorry?
I said physics.
Oh, yeah.
So the amplification step is the bottleneck.
Mhm.
And, you know, that kind of, we can push it down by a few more minutes.
Mhm.
But, you know, there's only so much we can push it down, because after a point, what happens is you don't amplify much. And then it takes so normally sequencing a read would take a few seconds, but then we are seeing like 8 minutes to sequence a target read, just because there's so few of them, because we are amplifying so less.
Mhm.
So if somebody develops really quick amplification techniques, like, you know, figures out how to even shorten that LAMP protocol, then yeah,
Right.
we can go sub-minutes, because we are pretty good at doing analysis and sequencing and making that fast.
Right.
Also, the nanopore is doing some more wet lab protocols, which can help these like binding, like catching. So there's a lead time of taking a DNA string and attaching it to the pore. It takes
Yep.
quite a bit of time. And they are doing some improvements to make that quick. So I guess if these two things come together, yeah, we could get down to minutes.
I see. So it seems like the big issues for this specific application has to do with biology,
Right.
and, you know, the hardware of the nanopore, right? The nanopore machine and things like
Yeah,
that.
yeah.
And that's really the bottleneck right now.
Right.
But maybe to the extension of that question is, are there other problems in the genomics, proteinomics areas that you've been looking into that still have a large computer architecture component that, you know, could be an interesting target for some people to look into?
Yeah, no, absolutely. I think population genomics is an exciting field. They are trying to use graph neural networks there, and to kind of fetch all this data out of the databases and then do the processing. That's interesting. Building just, you know, another thing which we have been looking at is kind of understanding the microbiome of your body. And many times they just sit there. They don't infect you. They just colonize you. But a trigger comes and then they start infecting you. So if you could predict that.
Oh, I see. So now you're talking about biological simulation of the microbiome, and okay, so that's really, or you know, I'm drawing conclusions here about simulation, right? But okay, maybe it's some sort of simulator. Okay,
well,
that's really cool.
Support tool, like, you know, gathering all this data, and yeah, so simulation could be a part of it, but yeah.
It looks like, you know, it's interesting how you went in and you said, okay, the nanopore techniques, right? They're doing the base calling, right? And then they give you the answer, and you're like, you know, we're just going to bypass that part and, you know, take the squiggles, and then, but if you go to the Illumina machines, you know, you don't have that ability.
Yeah, it's
right?
not
Because it's all locked up, right? It's a closed system, closed source, right? And so actually the nanopore system is than traditional systems, right?
Yeah, absolutely.
Now, I guess my question is, you know, you can take that to the extreme, right? Because now we've seen people build homemade, so of course homemade PCR has existed, but homemade genetic, you know, if you want to build your own genetic sequences, okay, now you can use a homemade system and do that, right? And so people are sort of, you know, building their own systems. And then on the flip side, we need software components to do all this, right? And things are still locked up because obviously these companies want to make some money. So I can understand why. Do you see this as an issue? Are there large impediments to doing this kind of end-to-end research because of restrictions? Like, I guess my point here is you couldn't have done what you did. First of all, you wouldn't have been able to get the timing, but that's fine. But just simply because the Illumina machines aren't open, you can't do that, right? You can't take the data, you can't do all the processing. They just give you the end result and you're done, right? Yeah. So are there other, I guess my main question is everything closed today, or are things moving more open? You know, what's happening right now with the situation?
So, I think of, so Nanopore is open software. Most of it is open. You can get a developer license and look through their code. And that's nice. And the reason it is open is because it is real time. So they have a feature to, stop and give a feedback and reverse the reads and so on. And because they want software developers to utilize that feature, they have made it all open source. So, that's cool. But yes, the hardware, the sequencer itself is all like, we are totally dependent on them. When they upgrade their system, when they upgrade the ADCs and all that, that's when we get the next version. Open sourcing that would be nice if you are a devices person and you can work with sensors. But I think just open sourcing the software itself is, that gives us that does leave a lot of fun stuff on the table. Like
right.
Nanopore has high error rates, right? And that's an unsolved problem for cancer. Like the way Jack, our group is solving is we just do so much sequencing to have a lot of depth and get around it. But, developing a software which can actually do true variant calling for oncology is a challenge, open challenge. So there's enough things to do in algorithm software and hardware acceleration.
So there's a lot there. It seems like we have a number of PhDs and professorships on the way, given your presentation. Okay, so I think we're a little bit over here at time. So what I'd like to do is thank Ritu again for all the great presentation and all helping us to answer all our questions. Thanks so much for your time.